Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Okay, we'll get started. It's an honor to have Eric Grimson here today. Eric is Professor of Computer Science and Engineering at MIT. He holds the Bernard Gordon Chair of Medical Engineering there, and he's also a joint appointment as lecturer on the radiology in the radiology department at Harvard Medical School and Brigham and Women's Hospital. So he has his feet squarely in medicine as an application area. Since 2005, he's been serving as the head of the EECS department at MIT. As far as some background, he received his PhD at MIT in 1980. He's a member of MIT's CSAIL group. I won't even have to say the acronym around here. You know what that means. And his group has done quite a bit of fabulous work in systems for recognizing behavior, for modeling sites, doing other kinds of computer vision applications, including object and person recognition, image database indexing, image-guided surgery, and so on. He's a fellow of the AAAI, fellow of IEEE, and was awarded the Bose Award for Excellence. I was joking that he had the Bose Award for quiet traveling across the country, but his excellence in teaching in the School of Engineering at MIT. Eric.
Thanks, Eric. Thanks. So, as has been explained to me, you're the studio audience, and the rest of the audience is out remotely. So this is going to be an interesting process for me. I have to also tell you that my feet are attached to my tongue. Those of you who had me in 6001 know this. I can't talk unless I'm pacing, so I will try not to pace too much. I wanted to do two things today, and one of them wasn't advertised in the talk, but I know there are some MIT alums here, and there are some MIT alums more broadly. I actually want to spend about 10 minutes. 5 to 10 minutes telling you about how we've changed our curriculum. We've actually changed it fairly dramatically. I take the slings and arrows in this all the time. 6001 is gone. It has reappeared in other places, thank you, Jim, but it has reappeared in other places. So I want to highlight for you how we've changed the curriculum and why, because I think it has impact on how we're thinking about the future. And then I want to spend about 25, 30 minutes, leave time for questions, to talk about how life sciences has become a major part of the research in EECS at MIT and where we see it as a point for the future and why we want to push at that. So some initial numbers, just to set the stage. Those of you who are alums know this. We remain the 800-pound gorilla on campus. We're still the largest department there. We have 700 undergraduate majors in the department. That's just under a quarter of all the undergraduates at MIT. At the height of the dot-com bubble, we were almost 40% of all the undergraduates at MIT, and we were dying. So it's nice to slim down a little bit. 700 undergraduate majors, 800 graduate students. That is about 125 MN students. The rest are doctoral students. As you can see, we graduate about 100 PhDs, a little over 100 PhDs a year. And we have, depends on how you count it, but basically about 122 faculty members. We just completed our hiring season and we've added 3 more people and we have 1 departure, so we'll be at 124. This is down slightly from our peak. I'll observe to you that there's some interesting numbers here. I point out to my dean regularly that, I've got 24% of the undergraduates, I've got about 15% of the graduate students, and only 13% of the faculty at MIT. And some additional resources would be nice. And the dean fairly regularly just reminds me that we're incredibly efficient and to shut up and stop asking for more money. So we'll do that. Exactly. I had a joking slide that I pulled out from here about how we teach. But I want to say a little bit about what do we teach our students. Because we just went through an interesting challenge, and I'll, with due respect to my colleague from this wonderful school, Stanford, I will point out that at MIT, our department gets rated 3 different ways in U.S. News and World Report, which, by the way, department heads never look at. We never actually look at those ratings, and if you believe that, right. We're rated as a computer science department, where we've been rated or tied for #1 for the last 15 plus years. We're rated as an electrical engineering department, where we've been rated or tied for #1 for at least the last 15 years. And we're rated as a computer engineering department, where we have been rated #1 or tied for none for the last 15 years. So one of the questions could be, A, why would you ever be department head? You can only take the department in one direction, which is to #2, which is a very bad sign. And B, why would you ever mess with your curriculum if you were actually doing something well? But we actually thought we wanted to address that issue. So the question is, so what do we think we actually teach our students? These are obvious statements, but they're important ones, because you notice it doesn't talk about content. It talks about thinking skills, how to think analytically, how to model complex systems, and I should have highlighted especially how to reason about them under uncertainty, how to transfer tools from one domain to another, how to communicate, how to work in teams. And what we actually basically said was we really wanted to focus on the transferable skills. And the reason for that is that when we look at where our graduates go 5-10 years out, 15 to 20% of them are doing something different than what they trained in. They're doing something that is not what they learned. So it's really important to teach them how to think so they can take those tools along. And it fits with the tagline that we use with all due respect to Intel for our department, which is that EECS is everywhere. We think that the tools that we teach you, the ways, the modes of thought that we help you understand are actually going to be critical when you do problems in a whole wide range of areas. And we're going to talk about life sciences today as one particular example of that. So we then took the challenge to say if the EECS is really everywhere, what does it say in terms of what we should teach? And we think that this both deals with what happens inside the department and what happens outside the department. So one of the questions I often get is, so why is it why is it an EECS department at MIT? Why don't we split? And there are a couple of reasons. One is it's nice to be the big department. You get a little more oomph. But the bigger reason is that we think that there aren't boundaries in areas. And so my two favorite examples, and I actually pulled this last night at a talk. I won't pick on a volunteer from the audience, although I see Zeke sitting there and I recognize him. You're a computer architect. Is that an EE problem or a CS problem? And the answer is hopefully yes. You know, you can't do one without understanding the other. You're a communications expert, a network expert. Is that an EE problem or a CS problem? Again, hopefully the answer is yes. So these problems don't have boundaries. We want to take advantage of that. And the second piece is that we think that this is really, as I said, a substrate for tackling problems in other domains, that the foundational skills you learn here are going to be crucial for problems in finance, for problems in life sciences, for problems in material science, for problems in a wide range of areas. So we want to focus on how do we build those. So I'd like to show you then how we changed the curriculum. And I'm going to both give you some high level and a couple of examples. We sat down with a group of faculty 6 years ago and had the faculty, about a dozen faculty, spent 2 years asking the following question. What should every good student in computer science or electrical engineering or both know? We put together a list. We looked at it. We concluded it was about a 7-year degree. We also concluded that most parents would not pay for the 7 years of a degree. There's simply too much to jam into a degree. And so one of the things we then said was, okay, we need to give our students more flexibility, more ability to decide what it is they want to know, while building a foundation that they can use to actually be able to go any direction. The second thing we decided is that it would be really valuable for students to see the same idea multiple times. Perhaps some of you had this experience. You studied really hard for that compiler exam. You knew all the ins and outs of the compiler, and as soon as you walked out of the exam room, there was a core dump, and all of that stuff went out of your mind, and you never remember it again. We wanted to cycle over the idea so that a student would see the same fundamental ideas multiple times through the curriculum. And the third thing we wanted to do was we discovered, maybe some of you experienced this, that it took us too long to get to the fun stuff. You spent 2 years learning all those foundational things before you ever got to what you think of as the interesting stuff. And so we wanted to create, I called it hands-on experience. A colleague last night said we should really call it context-based experience, but let students see things much better early on. So let me show you what the curriculum looks like. We have 3 degrees. I'm just going to focus on the one that most students do. There will be a quiz at the end, so pay attention here. But 3 degrees we do, and I want to show you just some highlights of the difference. So for those of you who are MIT alums, you remember we used to have 4 required courses, 6001 through 004. And if you were a CS student, you hated 002 and 003, because they were EE courses, and if you were an EE student, you hated 001 and 004. We've removed them, and students now take 2 required EECS courses. And I want to just briefly describe how we've changed both of them, but especially the first one. So 601 is a requirement for all students. It is an intro to EECS, and it talks about computation, control, circuits, and search. It is taught in a different way. I mean, one of the things I'm sure you remember is every faculty member likes to hear him herself or herself talk. And we have changed that. There's only one lecture a week in the class, but there are 2 required labs, an hour and a half software lab and a 3-hour design lab. And those labs are staffed by faculty. I actually have the pleasure of teaching one of those sections this term. And so what do I do in that section? I don't lecture. I spend the entire time walking around, looking over students' shoulders, kind of freaks them out a little bit, especially when it's the department head, but saying, you know, are you sure that that's the code that you want there? Or, you know, if you're stuck on this problem, what did we talk about? But basically doing one-on-one teaching. And what we're doing as well is having the students deal with all of the thinking in this class in teams, groups of 2, doing things on a mobile robot. So we went out and bought 50 mobile robots, made some company quite happy. And the students run a series of learning experiences on the mobile robots. I'll describe 2 quickly. The first thing you do is, what they're supposed to do is to get the mobile robot just to run down a hallway without hitting the walls. Easy, right? Well, maybe not. In fact, the walls in the lab now are all coated with bubble wrap. So we stopped chipping paint off the sides of the walls because you discover when you try and do that, oh, I need a feedback circuit. I need some control to make this happen. We walk through a whole series of things that culminates with the students actually getting the robot to run through a maze, where they do some circuit design. They design a little Lego head to fit onto the robot. They actually have it target things as it goes along, and it passes all the way through. It's a dramatic change in how we deal with it. The second course, over here, 602, is about communication. And very simply, it has 3 parts. We call them volt by volt, bit by bit, packet by packet. Sounds like a Three Stooges routine of some sort, but it isn't. Volt by volt, sort of the obvious thing. How do you move information around inside of an analog circuit? Bit by bit, once I've gone to that abstraction, what does it mean to move it around? And then, of course, packet by packet, when I go up to a network, what does it mean to move it around? Students build a software radio as part of this course in order to get some experience in how do you deal with all of that. Once students are past that, you'll notice that we cycle through a set of stages where they're going to see multiple paths through the curriculum, but students get to pick the path they want to go through. So if you're really a systems person, you follow a systems track through the course. You'll get a little bit of AI along the way, you'll get a little bit of theory along the way, but you'll be able to really focus on seeing systems, and vice versa. I want to point out one other change in our curriculum, which is way up at the top. It's called 6UAT. I know some of you took that. I have to tell you, when I helped design the course about 7 or 8 years ago, students hated me. Students still hate me. It's part of the job, but for a different reason. So actually, anybody here take 6UAT? So we'll see how well I do on this, right? Maybe you were one of the people who hated me when I did this too. What's 6UAT? It's about communication. The Institute changed its policy several years ago when we realized that most alumni told us that communication was crucial to what they do in their career, and we sucked at teaching it. They didn't actually, well, I'm on tape as I say that, right? But they didn't actually use that term. They used other terms that related to it. So the Institute said you need to add some communication courses. Most departments basically took existing courses. Lab course, you write up your lab report. We decided to bite the bullet, and we said, if you're going to do communication, you need to really learn how to communicate. So here are the things, you'll see if I do this right, that you learn in that class. You learn how to do an elevator pitch. In fact, it's a wonderful example, right? The example is you're in an elevator with Bill Gates. You've got 3 floors. How do you sell them? You learn about how to put together a PowerPoint presentation. You learn how to model your audience to think about how well am I communicating with them. And I don't know if they still do it, but we certainly did this early on. One I loved, how do you send email without annoying the recipient? A useful talent to have. Culmination of this is, right, you all got to do a presentation on your research orally. And I don't know if when you took it, it was this way, but certainly now the presentation is not to MIT peers. It's to high school students from Cambridge who come in. So you really have to build a model of this. So as I said, students hated me. We won't take a poll here of the group that put their hands up. But initially, students hated me because it was like, this is soft stuff. I mean, this is, what is this, Harvard? We're not going to do this. Now students hate me because they say, why do we have to wait until we're seniors to take this class? Because this is really valuable if I want to get that internship at Microsoft or at Intel or at, I almost said the Mountain View World, that other company in California that also takes on a lot of interns. We're trying to react to the fact that students need to be better prepared to move into a career and thrive in it. And so changing 6 UAT was one of the things we did there. We did retire 6001. And yes, I took lots of hate mail over that. But we put a lot of the ideas of 6001 in other places in the curriculum. But what you see here is a much more flexible curriculum, a curriculum that focuses on transferable skills and modes of thinking, and one that really tries to blur boundaries. Okay, the other piece I wanted to talk about is another change that's happened in the department. And I want to give you the number here to set the stage, and then I'll show you some examples. So we have about 120 faculty in the department. And I did a count about a year ago of how many of those faculty members had a significant research presence in the life sciences, broadly construed. And the answer was about 40. So 1 in 3 EECS professors does something that's life sciences related. And we think that's a trend that's going to continue as we see more and more opportunities to use EECS kinds of techniques in the life sciences. It's also driven by the students. Students come in with much broader interests than what they did in the past. And often the interest is not discipline-driven, it's problem-driven. I want to tackle environmental issues. I want to tackle transportation issues. I want to tackle healthcare issues in the developing world. And so we're interested in seeing how can we build those bridges. So what I wanted to show you was a sampling with a little bit of depth, since this is, after all, a talk to a bunch of technical people, of things that are going on in the department. Now, I'm not going to do all 40 of these, although I will show you the names at the end of everybody doing it. I'm going to take about 3 different areas and show you some depth of the kinds of things that we're doing in these areas to let you see how EECS ideas are having a real impact in this area. And I know it's a topic that Microsoft knows, because there's a bunch of people here doing exactly that. But let me show you our version. And I'm going to start with the area I know best, which is work that my own group has done and my colleague Polina Goland has done. And I'm going to start, I should have warned you, gross pictures coming up. I'm going to start with a simple example. If you have a brain tumor up here, close to motor cortex, so it's pushing on the part of your brain that controls movement, you'd like to get it out, obviously without, or you'd like the surgeon to get it out without cutting motor cortex. And what does a typical surgeon do? Well, they're going to take an MR scan like this. The tumor is going to show up. It went by a little quickly. It was right about there. A surgeon will take an MR scan. They'll get those slices of the MR in. They'll identify where the tumor is. And then in conventional surgery, they'll actually open up, cut a big skin flap out, take a piece of your skull out, expose the cortical surface. And then over on the far wall, they may have those MR scans there that they're looking at. You're here. And at this point, you need to pray that your surgeon was a math major. Why? Because they better be really good at geometric transforms, because they're looking at the wall to see where the tumor is, and then they're going to root around in your brain with a spoon. I don't know about you, but I'm not thrilled with this idea. So we said, wouldn't it be nice if instead you could turn neurosurgeons into Superman or Superwoman? Give them X-ray vision so that they could look at a patient and see through skin and bone to see exactly where the structures are beneath. And in particular, so that during surgery, as a surgeon inserts a surgical instrument into a cavity, in real time you could see exactly where the tip of the instrument was relative to a 3D reconstruction of the patient, and in real time you could update the MRI images cross-sectionally to see exactly where the tip of the instrument is, so you know what's around and how you get to the target. I'll note to you parenthetically, by the way, that in my experience, at least with Boston surgeons, every time I've suggested to a neurosurgeon, I can turn you into Superman, I get a quizzical look and a suggestion that they don't need any help in achieving that status, but it's kind of fun to do it anyway. And now I've just annoyed all of my surgical friends. So to show you an example, what we want to do is use computer vision and machine learning techniques to build very detailed, patient-specific reconstructions of anatomy. That giant green thing there is a tumor. You see the ventricles of the brain. You see the major blood vessels. You see the cortical spinal tract that controls movement. And up at the top, you see functional cortex. And by building those models, and I'm going to show you very briefly how we do them, it allows the surgeon to both plan and navigate during surgery through minimally invasive openings to execute out the surgery. So I want to just give you a quick highlight of some of the tools we use here. So basically, we're going to build patient-specific models, add some additional information, register it to a patient so that you can do augmented reality visualizations, and then adapt to changes in it. And these are examples of the kinds of models we build. We've actually run our system on about 1,000 surgical cases at Brigham and Women's before we retired the system. So it shows you the examples. And while we certainly do neurosurgical cases, we can do things like tumors in the spine, in the throat, knee surgery. We've done a wide range of these kinds of things. One of the reasons it's handy is it lets surgeons visualize particular targets. And so right here, this little thing that spot right there is what's called an AVM. It's an arteriovenous malfunction. It's a place where an artery and a vein in the brain have grown together, and you need to be able to separate it out. So it lets the surgeon visualize. We can do quite complex reconstructions. So this is a CT scan of the middle section of a subject. And just using some level sets methods, if you've seen those, we can extract out very detailed reconstructions of, in this case, the aortal cavity, which we can then measure for things like width of the tissue in order to be able to do measurements on clogging in those tissues. One of my favorite examples is to do things like a virtual colonoscopy. Colonoscopy is not a fun thing, as I can attest, having had one done recently. And so one of the things we wanted to do is to say, what if we could just do a virtual colonoscopy, take a scan of the subject, and then fly a virtual camera through the colon, looking for places of high curvature. So there's a polyp right there that just went by in blue. And so this is an example done by an MN student several years ago of basically doing a fly-through of a reconstructed colon. Part of the reason I like this is I point out to my students, if we ever have any trouble with NIH funding, we can probably make up for it in games. I mean, this just screams, gut revenge or something, right? You've got to be able to make a video game out of this somehow. But it gives you an example of the detail with which you can build these kinds of reconstructions. So let me show you very briefly, and this is the one example I'm going to do in detail, and then I'll move on to some other ones, how we do this. Because it captures many of the tools that I know you use here in terms of statistical methods in machine vision and machine learning to do it. So we want to simply build a model of tissue appearance and then add in some information about expected shapes in order to deal with it. So a tiny bit of math. The idea is we get a scan, an MR scan, we want to label it at every voxel with what kind of tissue is there, and we do it by saying we want to find the kind of tissue that maximizes the expected appearance of the image given we know what kind of tissue we're expecting there. And so the easy way to do it is just to take a set of samples, do a mixture model, and basically do nearest neighbor to label them. And if you do that, these are a set of scans of a subject, happens to be a subject with multiple sclerosis, but each scan is taken about 6 weeks apart. You look at that, you can say, I can see where the different tissue types are. And if you actually label it that way, you get this. Now notice, in the space of 6 weeks, this patient's brain went entirely from gray matter to white matter. This seems pretty unlikely. So there must be a flaw in this, and this is where the modern techniques come in. So the problem is that you're caught between Scylla and Charybdis, or if you were not a humanities major at Harvard, a rock and a hard place. All right? So what's the problem? The problem is that these images have what's known as a nonlinear gain artifact. That is, the intensity of the magnetic field shifts, and therefore the same kind of tissue in different parts of the field will give you different intensities back. The reason you're caught between a rock and a hard place is if you knew what that gain field was, you could just correct the image. You could normalize it, and then you could do these standard statistical methods. But you don't know what the gain field is. Well, if you knew what the tissue types were in the brain, you could predict what the intensity should look like, compare it to the image, and figure out what that gain artifact was. And of course, you see you're caught in this wonderful loop. And the reason I like to raise this is, to my knowledge, it's now an obvious thing to use EM on it, but this was done actually almost 20 years ago and is the first place in computer vision, I think, that EM actually appeared as a solution. And it's a nice example of why a wonderful statistical technique will let you iteratively solve for both of those. And so that's what we do. You turn it now into an EM problem where you're solving for the hidden parameters of that gain field. And I'm not going to go through the equations, but there they are. It simply says, I've got a weighted equation that says it's a Gaussian distribution shifted by the gain estimate, normalized by a prior that's initially just the overall ratio of tissue types in the brain. I'm going to fix that in a second, with a normalization factor underneath it, and then a maximization step that just optimizes those things. So if you've seen EM, this is a standard kind of EM equation. And now I go instead of from there to there. So it does a great job of picking it out. Now, it turns out that there are actually still some subtle things it won't find. If you look carefully right here, you'll notice it's slightly darker. That's the amygdala-hippocampus complex in the brain. If I told you that's where to normally expect those structures, it's a lot easier to take this and that and decide how to draw the boundary. So an example of a second nice statistical method we want to add in is, okay, how do we take advantage of that? And we did this the following way. We took 50 normal subjects. I have to say, by the way, I love that term, normal subject. I don't know what it means. I will simply tell you they happen to all be Harvard students, which worries me slightly about how normal they were. We had no MIT students there, but we took 50 normal subjects. You pick one of them as the canonical one, and you use a machine vision algorithm to elastically warp all of the other images so that they fit that one. So that says each one of those red arrows there is a deformation, a transformation that warps them in. For each one of those subjects, you then run a segmenter, and you pull out where are all the places where there's white matter. And then you apply those transformations to align them up to a common framework, and you count. And it builds you a probabilistic atlas of where to expect to find white matter. And now you go back to, or you need to align that to the subject. You do that just with a pair of transformations. I'm going to skip by that. And you can now add that in as a prior that says, I know where to expect white matter. It's going to happen in particular parts of the brain. The other thing we do is we say, we know that different structures have particular shapes. The corpus callosum is always shaped like a potato chip, for example. And I want to take advantage of that. So I'll spare you the details, but we also build a set of geometric models of expected shape, and we add those into one giant EM segmentation method. And now we can solve automatically for what is the label map, the registration, the shape that maximizes this probability. And we get now segmentations that can pull out very subtle structures by automatically co-registering and segmenting the structures. We do this hierarchically. It's a standard CS trick, and I just want to show you the example. We now segment out 51 different separate structures in the brain. So you build a very detailed model of that brain. And that you then align to a subject to do image-guided surgery. So point of this first example is that it lets you see how standard statistical tools, or at least standard these days, are really valuable in letting you take computer science ideas and build much better surgeons. Let me show you the second example here, also in the brain context. A tiny little bit of a neuroanatomy, although I know there are some course 9 people here. Axons of nerves come with a particular structure. They're organized in these nice fiber bundles. And one of the properties of them is if you take a sequence of MR scans very quickly, you can measure what's called a tensor at those points, which basically tells you how likely is water to move in different directions. Now, why in the world should you care about, and maybe you think that most faculty have water on their brain, but why should you care about how water moves? Well, a water molecule is going to diffuse in particular directions, and if I happen to have fibers, like axon sheaths, it's going to want to move along the axon, not across it. And so in particular, if I take that tensor information, I can decompose it to get a sense of the local orientation of tissue everywhere. And in particular, I can stitch it together to pull out the white matter tracks. These are the fibers that connect up the different parts of your brain. And in fact, you can see here a computer algorithm doing it in a comparison to a dissection of a post-mortem brain. Okay, why would you like to see that? Well, once you have those tracks, you can measure properties along them, like how strongly are they oriented. Don't sweat the math. This is just simply saying, given those 3 eigenvalues, I can tell you how strongly things are oriented. And that allows you to do these kinds of reconstructions. I happen to love this picture. It's done by my colleagues at the Brigham. The joking version of it is, there's the wiring diagram of somebody's brain. It is literally all of the fiber tracks that are connecting up the different parts of the cortex. One of the things that's fabulous about this, by the way, is if we find a tumor in one part of the brain, we can have computer algorithms automatically trace through what is it connected to decide what other things might be implicated by it. But let me show you one other example of how we can use this, which is those weird things I went by really quickly about the local texture orientation of the brain. We can measure that fractional anisotropy, and we can detect when it's changed. So this is an example of a subject with MS. This is the corpus callosum. That's the sheath that connects the 2 halves of your brain together. And you'll notice that it should be strongly red because it's a very strongly oriented thing, and there's a gap right here where an MS lesion has actually kinked the fiber, if you want to think of it that way. So we can measure the FA, and I want to simply show you some examples. Given those tracks, we take a set of machine learning algorithms to do clustering on them, to pull out the distinctive clusters that are individual tracks within the brain. And along them, the second thing I should have said is, and we do this for 2 different groups of patients. 50 normal subjects, 50 subjects that are schizophrenics. And we ask the following question: are there differences between those structures in those 2 populations? And so what you're seeing here is 3 different parts of the corpus callosum. The blue curve is the fractional anisotropy, how strongly oriented it is in normals across all 50 subjects. and the red is schizophrenics. And notice some interesting effects. There are very subtle but statistically significant changes in particular parts of the corpus callosum between those two subjects, or those two kinds of subjects. Do the same thing, for example, on the cingulum bundle. And in one of my favorite ones, in the cingulum bundle, we can also notice that there are differences between the left and right hemisphere of the brain in normals and in schizophrenics, but it is accentuated in schizophrenics more on the left side than the right. So what's this doing? It's computer science tools that are letting neuroscientists see very localized effects of changes in anatomical structures. Right? Let me show you one other example, again from my colleague Polina Goland. We can ask the same question about anatomical structures, not just about fiber bundles. And so one of the things that we can ask is, if you're given two sets of example shapes from normal subjects and from schizophrenics, is there a difference? Now, I'd suggest to you, if you look at that bottom scan, it's pretty hard to spot a difference. But in fact, it turns out that you can build a computer graphics, if you like, representation of the shape using what's called a distance map. You can put those all into a very high dimensional space, and you can apply machine learning tools to basically build two clusters. And you can then ask the following question, which is, if I give you a new example, what do I have to do to the shape to make it most easily look like a schizophrenic or a normal? And this is useful not only for classification, but also for deciding how do you have to change it. And in fact, what you find is that there are very localized effects. Like you have to compress here and you have to expand there in order to take this example and shift it. So this is giving you an example of a localized way to actually assess neuroscience changes. We've also done this again on schizophrenic studies. I wanted to show you one quick highlight of this. This is now looking at schizophrenics, people with affect disorder, and normal controls at 4 different structures in the brain. The charts you see at the bottom here are simply showing you the difference. I want to just point out this one here. This is left superior temporal gyrus. And you'll notice with schizophrenics, it has a noticeably different set of parameters. The reason I like this, and I need to be careful saying this because I don't want to make it sound like we have solved the problem. The reason I like this is, anybody remember the neuroanatomy? I know there's a course 9 graduate in the crowd here. Anybody remember what superior temporal gyrus does? No reason why you would. It's involved in hearing. What's one of the interesting attributes of at least some kinds of schizophrenics? They hear voices. Now, can I tell you this is where it's coming from? Of course not. I'm not that good. But can I tell you, boy, it's kind of interesting to think that you may have seen at least a correlate of some of the effects that the schizophrenics feel. So this is a place where, again, we're able to localize this pretty well. Let me show you one other example from neuroscience, and then I'm going to shift gears to a couple of other examples. This is an example, again, from my colleague Polina Goland, and it's to do functional parcelation. So here's the idea. They took a set of subjects and sat them down and had them watch a movie. And while they were watching the movie, they did functional MR recording. So if I just remind you, if you haven't seen it, functional MR is recording the blood oxygenation rate in the brain. It's trying to tell you what parts of the brain are lighting up or being active as you do this. They did some controls to compare it against. They took one of those segmentations I showed you of anatomical structure and mapped the two together. So at every point in the brain, they had what kind of tissue was there, and they had a time series of what the activity in the brain was. They put that into a really high dimensional space, and they basically said what parts of the brain are coactive, what parts of the brain are responding at the same time to this. And I want to show you what happens if you do that with just a k-means clustering. I should say, by the way, the movie they had them watch was The Good, the Bad, and the Ugly. And I would have expected just a flatline brain. I mean, why would you ever respond to that? But nonetheless, they did it. So let me show you the example here very quickly, which is the first thing they found was if you do the first k-means decomposition, the brain separates out, or the coactive areas separate out into 2 pieces. Parts of the brain that we know are stimulus dependent, visual cortex, motor cortex, somatosensory cortex, and parts of the brain that we know are involved in more introspective kinds of things. So not a surprise. If you do the next level, it breaks it out into visual and motor cortex, that they respond differently. Again, not a surprise. Motor cortex separates down into superior temporal sulcus and other pieces. And visual cortex separates out into retinotopic, that is parts of the brain that just have mappings that are like what the retina has, and then higher level vision. But notice what happens at the next level. And I'm actually going to flash back and forth. You can watch the change up there. That intrinsic area breaks down into 2 different subpools. And the neuroscientists don't know what they do. So it's an intriguing little insight into there's two different populations of cortical sheets that are basically interacting different ways. And similarly, the visual cortex, the high visual cortex, separates down into two other pieces, one of which we know, one of which we don't. And finally, motor cortex also separates out that way. And this is now causing our colleagues in brain and cognitive sciences at MIT to be really intrigued with asking questions about how could we test what those parts of the brain are doing. So again, an example of CS tools coming along. I'm going to spend about 5 more minutes just to give you some other highlights in a slightly more rapid fashion. And I want to now switch to micro-nanotechnology. So not a CS problem, but a wonderful EE problem. This is done by my colleague Sangeeta Bhattacharya, and I like pointing out Sangeeta because it shows you how my department has changed. So Sangeeta is an MD PhD. By the way, at my age, I like having an MD in the department in case something goes wrong. But she's also a Howard Hughes medical investigator, and I think that's a wonderful statement that an EE professor is an HHMI. And she's really interested in basically looking at the interaction between synthetic and living tissues at a range of scales. And I want to show you 2 examples. One of the things Sangeeta worries about is liver function, hepatocytes, and how liver cells interact. And one of the things you'd like to know is you know that those liver cells have to be close to fibroblasts in order to have the right kind of behavior. You'd like to know, well, how close do they have to be and over what time course? So she designed these wonderful comb-like nanostructures, which you coat with different cells. And you can intertwine them so that they're at different distances away. And you can monitor them over time. And one of the things that she was able to find is that, for example, you need to have the cells touching for a day, and then within 350 nanometers for at least another 3 days in order for the cell to operate properly. Now, why would you care? Well, if you're designing pharmaceutical solutions to things, it's really nice to know how the cells behave as you do it. Let me show you my other 2 examples of my favorite ones. One of the other things Sangeeta does is she plays with quantum dots. So quantum dots are these wonderful, well, they're obviously tiny dots if they're quantum dots. They have a number of great properties, one of which is that they fluoresce under ultraviolet light. Another one is that they have a very interesting volume to surface area ratio. So one of the things that Sangeeta has done is to take those quantum dots and to grow proteins on the outside, so ligands on the outside, and specific ligands that are attracted to tumor cells. So you create these tiny quantum dots, you put them in a fluid, a diffusion, you inject it into the bloodstream of an animal. Doesn't do much, it flows through, but when it gets close to a tumor, the ligands are attracted to the tumor. They attach, and you basically aggregate all of those structures around the tumor. When you've done that for a while, you take a UV light, you shine it on the animal, and the tumor glows. You can literally see it. So it's a wonderful way of sort of isolating it. The real reason she wants to do it, and work that she's now carried on, is you can also construct those quantum dots so that they will aggregate together, creating a cavity into which you can put a pharmaceutical. And again, you attach to it a set of proteins or ligands that are, in this case, heat responsive. You put that into the animal, it flows through, it attaches to the tumor, and then you simply irradiate that portion of the body with a heat source. Those links break, it leaves that quantum structure behind, and it unfolds and does local delivery of the pharmaceutical right on the spot, which is a wonderful sort of example of very localized therapy for treatment. I want to show you one other example from, again, the EE side, just to show you, well, let me show you one other example. This is my colleague Fatih Yanık. Fatih was really interested in understanding how nerve cells regrow, or under what conditions do nerve cells regrow. And so his way of attacking this was to start with this worm called C. elegans. I never remember what the C stands for, but it's a worm that physiologists use. It has the property that it has neurons that are easy to get at but look a lot like human neurons. And so Fatih wanted to know, could I clip an individual neuron and then watch it regrow? So he did the following thing. He built a microfluidic device that takes an individual worm, isolates it, immobilizes it. He does surgery with a femtosecond laser. And the advantage of the femtosecond laser is it'll clip the neuron, but it's such a brief flash of energy that it doesn't heat up the tissue. So you don't do any damage to the tissue nearby. So he literally clips the neuron, and then he bathes the worm in some range of chemicals, and you watch to see how the neuron regrows, which it actually does. And he was able to identify, in fact, a chromatin remodeling factor that enhances neuron regrowth by a significant factor 5-fold. The other nice thing about this is a wonderful sort of high throughput device. Fatih has done surgery on 30,000 worms. I got a letter the other day from the SPCW, the Society for Prevention of Cruelty to Worms, very unhappy about the fact that we were doing this, but we're going to do worm surgery. But it lets him do very high throughput things to actually understand how these processes work. He has done the same thing with a little bit higher order animals. There is a rumor around MIT that there are a lot of faculty who are worms. I know you've had experiences with many of them, but we do have some vertebrates among our faculty. So he did the same thing in this case now with zebrafish, where he builds a wonderful little microfluidic device that grabs a zebrafish from a reservoir, captures it, orients it, re-immobilizes it. He does femtosecond laser surgery on it, and then does 2-photon microscopy to observe how things change inside of that process. And so this allows us to see ways in which you can actually start to orient or understand nerve regrowth in a range of things. I'm going to skip by that and do one last example from my colleague Colin Stoltz, who does molecular dynamics modeling. And I want to show you a particular example here. So Colin is really interested in collagen, which is about 30% of our body. Among other things, it forms cartilage, which is really important to people like me who have bad knees. It also is in the heart. And in fact, one of the standard causes of serious heart attack is, as you have irritation in the heart, collagen forms around it to try and isolate it. If the collagen breaks, you have a disruptive event. You have a dramatic heart attack. And one of the things Colin did was to do very high-scale molecular dynamic modeling of the energy states of collagen. And in particular, what he observed is that if you do these simulations, you'll find that there are some collagen-like peptides that look a lot like collagen. In fact, they will interact with collagen, but they have a hidden cleavage site. And if you unwrap these, or if you let them interact, you'll find that they will unfold in different ways. So standard collagen has one kind of unfolding. These mutants have a different kind of unfolding. And in particular, what you can find is that if you model those thermodynamics, you find that near the cleavage site, you have the opportunity for those mutants to unfold in a way that then interacts with the rest of the collagen to cause damage to the collagen. And that allows you then to actually come up with ways to decide under what conditions are you more susceptible to a heart attack, and under what conditions can therapeutics actually change that distribution of the collagen in order to be able to decide, for example, what collagen-like peptides will mimic those regions and what other ones will actually allow you to control where those structures go. This is a wonderful, very large-scale simulation problem in which you're simulating literally the molecular interactions of very large-scale structures in order to understand what is going on inside of them. I'm going to skip by this last one. I want to just show you an example of dealing with heart attacks and simply say, I wanted to give you a quick overview, but here are the other areas around the department in which we have life sciences interactions. Synthetic biology, medical sensors, low-power devices, prosthetics. And by prosthetics, I mean actually not limbs, but artificial ears, artificial retinas. Obviously, bioinformatics and computational biology, signal analysis, ferrofluidics, biophysics, and speech and hearing. I started off by saying about 1 in 3 faculty members in the department does EECS things in the life sciences. And you can see at least an example of where all of those pieces show up. And I did promise to leave time for questions. So, with that, I'm happy to take any questions you have. Those could be curricular questions, or those could be EECS questions, or those could be, are the Red Sox actually going to come back this year? Please don't ask me that one, although we won last night. Questions? Bob.
I was fascinated to see that you make such heavy use of EM. Just before coming here to hear your talk, I was talking with one of my colleagues and our summer intern about how we're going to use EM to solve a problem in statistical machine translation.
Yeah.
And one of the, in our applications of EM, one of the problems we're constantly running into is we're always trying to run EM on problems that are too complex and the optimization surfaces are not convex.
Yep.
Do you have that problem too?
Yes.
Do you have ways of getting around it?
We have, I think,
you showed beautiful results.
So we do have that problem. I suspect we don't have it as badly as you do in things like natural language. We don't have bulletproof solutions to it, but we, like everybody else, find heuristics that get us around it. I mean, I was going to make one other comment, and it's an obvious one, I'm sure, to this community, but I think it shows you the change in the field, that statistical methods are so much more important. So the comment is, about 4 years ago at MIT, we had a task force that asked the question of what should every undergraduate at MIT know when they graduate. For those of you, I know there are alums here, that's the GIRs, the General Institute requirements. So at MIT, it's 2 physics, 2 math, chemistry, and biology, 2 rest subjects. The reason I'm saying it, Bob, is that one of the things that we found when we went around in this task force and asked faculty in every department at MIT, engineering, science, political science, economics, there were 2 topics that regularly came up where everybody said every student at MIT needs to know those 2 topics. Anybody want to guess what the 2 topics were? Sorry? Statistics. Well, yeah, statistics and probability was one. The other one was computation. You know, and those were sort of nice to hear. Now, unfortunately, we didn't manage to change our GIRs for political reasons, but it was striking that, right, those have both risen in some sense to be equal partners in the STEM array, if you like. And so, yeah, I think that the statistical methods are just crucial for everybody to know. It doesn't matter what you're doing, you need to know them. So, yeah.
On the prosthetics, you talked about retinal implants. I'd like to hear more about it, but I was wondering if on the horizon of the department there's direct brain connection in and out.
Yeah, great question. So let me try and quickly answer both of them. So the retinal implant work is the work of John Wyatt. They have been designing for actually probably about 15 years small array implants that are biocompatible, that go into the back of the retina to provide, it's a low resolution, but I don't know, on the orders of 10 by 10s of arrays inside of the retina. They have now gotten to the place where they are doing animal studies, which is actually quite exciting. My guess personally is that it's going to be a much longer haul than it is in auditory. So cochlear implants have been around for a long time. It's actually a nice MIT invention, but they're actually pushing pretty hard at that. The direct, brain implant things, we don't currently have faculty doing it, but we have a lot of faculty getting interested, and there are a number of great colleagues around that are doing this really well. So it's something that I think the department will see in the future, is that we're going to start seeing more of that kind of thing. Again, I'm not certain how long it's going to take, but I've seen some really nice work. Michael Black at Brown in particular, I know of, has done some great stuff, but there are other people who've done this really well. It's fascinating stuff. Other questions? Yeah?
Can you talk more about the study you did with the hospital? You mentioned you had about 1,000 subjects. What did you do with each one of those?
Yep. So the example, this was in the neurosurgical case. We built basically a surgical guidance system that would allow surgeons to use this visualization technique to do neurosurgery. We hoped more effectively. We actually ran the system on 1,000 cases at the Brigham and Women's Hospital.
That includes the augmented reality?
The version we used did not use the AR stuff for reasons I'm happy to tell you about in a second. It used basically a monitor right in front of the surgeon in which he would see, so the orientation wasn't a big problem, in which he would see the tip of the instrument in cross-sectional views of the MR volume as he went through it. Come back in a second to why we didn't do the AR version of this. But we did run this system, like I said, for 1,000 cases. What were we looking at? The outcomes we were interested in was, what did it do to time? And what did it do to accuracy? And what we found from the surgeons is that it cut the time of the surgery down by a factor of 2. Surgeries that used to take 8 hours, they now did in 4. For an interesting reason I never thought about, and I'll simply share with you, which is neurosurgeons at least pointed out to me that a lot of the time of those surgeries is getting to the target. I mean, you sort of think about it, right? I mean, you got an abdominal thing, so you move a little tissue around. Okay, so I could use it. It could be a little thinner. In the brain, moving a little tissue around, you're moving something around you don't want. So getting to the target really fast, much faster is a big plus. And that was one of the big changes. The second thing was, and I didn't show it here, what we found was that the surgeons were now taking on surgeries that they would not have done in the past. Typically surgeries where the tumor was close to a piece of cortex that they could not identify visually and they did not want to damage. So the most common surgeries we did were actually surgeries up near motor cortex, because we could show them where motor cortex was so that they would avoid it. So why did we retire the system? My students got really tired of getting up at 3 o'clock in the morning to go over to the surgery. And we had done this study. We wanted to move on to other things. The other thing that was nice about it, though, is we always clearly worried about negative side effects, negative outcomes. And I can tell you we had zero. One of the examples I showed you, sorry?
Who's productizing?
There are a number of companies who will now do this. Medtronic sells a system, GE sells a system. There are companies that will now do it. And in fact, I'm going to come to this question in a second. The most common, and this was just a fun stat to have, the most common place where you use these kinds of guidance systems is actually in sinus surgery. And I was surprised by that until a surgeon pointed out to me that sinus surgery has the second highest rate of malpractice suits. And if you think about it, right, you're sticking a sharp instrument up a structure where all that's between you and the midbrain is a very thin membrane. So if you go too far, you hit the optic nerve, you blind the subject, you hit the midbrain, you paralyze the subject. So, there are things that you can do there.
Hold on, you're going to talk about the AR?
Yeah, the AR. So the reason we moved away from it was twofold. One was one of the questions is how do you show it? We actually tried goggles. Right? We put goggles on surgeons, so it was like a wonderful video game. At least at the time we did it, the surgeons hated it because the lag times bothered them. And, it's not great having a surgeon over a surgical cavity losing his lunch. I mean, it's just, not a pretty sight, right? So we thought about using monitors in front and having the surgeons see their hands in it. That one worked pretty well. We also tried actually directly projecting, just with that kind of projector, directly onto the patient. That's a wonderful challenge because the surface you're projecting onto is fluid covered, so it tends to reflect in funny ways. The reason I say all of it is I think you could come back to it, and in fact there are systems now that for microscopic surgery, if you're operating through a microscope, they do AR, in which you build these kinds of models, you pump them into the two ocular pieces of the stereo microscope, and they can literally see where the structures are below where they're... Sorry?
That's very clever.
Yeah, it's a wonderful solution, I agree. Steve?
I was wondering if there were some trust issues and FDA approval issues. I was involved in a startup right after I finished MIT, and that was they had some great technology. In fact, Steve Piper, who might have been involved in this, was there also. And these were some of the stumbling blocks at the time.
Right, so there are two pieces here. I want to do them both quickly. There are FDA issues, and of course, when we're doing this at the Brigham, we have to go through IRB approval both at MIT and at the Brigham to make sure that we're doing it under the appropriate controls which we need to do. But it is a case that to get the technology out that's a big stumbling block because you've got to go through the FDA correctly. You've got to go through the FDA approval to make it happen. The trust issues are an interesting. I'm not certain I know quite the spin you want on it, Steve, but I'll give you my version of it. I'll have to tell you that there were times when I would get a little panicky because the surgeon, who is a brilliant surgeon, I mean, people flew in from all over the country to have him do surgery, is a gentleman who's now in his 70s, and he is not tech savvy. And so it would worry me a little bit that he would over-trust what we were doing. So one of the things we did, for example, was to build in protocols. So we had things like, if I'm showing on a monitor where we claim the tip of the instrument is, the protocol was, as he's doing surgery, every time he got close to a very visible landmark, okay, we asked him to make sure that he would put a probe on the landmark so we could see on the monitor how far off are we. And if we got too far off, we shut down because, the trust is he thinks it's perfect. He kept saying, you guys are from MIT, you're great. And I said, thank you, but, if we get it a little wrong, there's no reset button on a neurosurgery case, so it makes it a little bit challenging. Any other last questions before we wrap it up? I know we're coming up on the end of time here. Any curricular questions? I'm happy to...
That last story just reminds me of a story from the AI lab. I think this was a couple years before you came, but you remember Peter Solovitz and Tony Gorey were doing medical stuff back then.
Yep.
And I heard Tony Gorey tell a story about how he'd flown down to Baylor Medical Center in Texas, and he met the great Michael DeBakey, who must have been about 60 at the time. And he said, DeBakey said to him, young man, I've been a surgeon for 30 years and I've never needed you before now. Why do I need you now?
Well, and I'll add as a last piece, especially for those of you who want to start doing things in the medical domain, it's an interesting challenge to get to. Among other things, the culture is incredibly different. You really, I know Eric has seen this as well in his own work. You really have to get used to it. My other experience there was that I found, at least in our case, the neurosurgeons divided into two roughly equal camps. There was the camp that absolutely did not want to proceed without the technology because they saw the advantages of it. They tended to be the more experienced surgeons, which was interesting. And there was another camp that basically didn't figure they needed us, and they tended to be the younger surgeons. I find that kind of interesting that the guys with a lot of experience sort of know all the things that can go wrong and why an extra piece of technology could help you out.
Three lawsuits is a great learning experience.
Sorry?
3 lawsuits is a great learning
Yeah, that's a good point. A couple of lawsuits probably gets your attention pretty quickly. And you
And
want
just as a point, counterpoint, Bob, I drove across from San Francisco to Australia with the baking in the seat next to me, talking about machine learning the whole way. He was really into it. So by that was 95, though.
Okay.
Yeah, so he had seen the light. That's great.
Years like
Good.
75 versus 95.
Yeah.
Okay, thanks very much.
No, thank you.